Welcome, everyone. My name is MJ. I am the APEC Sales Manager at Align Genetics, and I thank you for attending to How to Choose a Cell Counter webinar. This webinar will last about 30 minutes, and if you have any questions, feel free to contact us by email or through the website. We'll be glad to answer any request. First, let me share some information about Align Genetics. Align Genetics, formerly Logos Biosystems, was founded in 2008. We have a large network of partners worldwide with deep focus on innovation. We have two brands, GeneScale, which is an in vitro diagnostic point-of-care solution brand, and Logos Biosystems, which is already a leader in imaging solution for life science, including digital microscopes, tissue clearing solution, and cell counters. Today, we will talk about cell counters. Cell counting is necessary, but most of the time a tedious step for in vitro cell culture. But cell counting is really important. When you want to monitor the health of your cells, you want to check the proliferation rate, you want to assess immortalization, transformation, you want to check the transfection or infection, or anytime you want to seed cells for subsequent experiments, especially for cells-based assays. Therefore, it's critical that your cells are counted accurately with a reproducible approach, especially when you want to quantify measurements of cellular responses. And among the challenges, when we talk about cell counting, the first challenge is actually the cells themselves. You have a huge variety of cells available that you may want to count. It can be a bacteria, which can be 10,000 times smaller than mammalian cells, or you can have budding cells like yeast. Or even if you're working with mammalian cells, you don't always work with cell lines. You can also face the presence of debris or contaminants like red blood cells if you're working with the PBMC or leukocytes. Also, in the same sample, you can have cells with different sizes, and that's the case for MSC cells. Or you can have clusters or different shapes. And maybe you're interested in counting other kinds of cells like pollen, protoplasts, spores, sperms, ovocytes, microalgae, or parasites. So the diversity of the cell is the first challenge to face. But did you know that about 5 years ago, still 70% of the researchers considered that the hemocytometer was the gold standard for counting? As we probably all know, hemocytometer is composed of the mounting support, a cover slip. Then you load your cells and you're going to count them with the grid as a reference. This is a very old method which was created more than a century ago, and this is still used by many and many labs. But it has a lot of recurrent issues. The first issue is the perception of a cell. If you ask 3 different people to look at the same complex or messy sample, the number will all be different. And I'm not talking about pipetting errors, dilution, volumes, or maybe sometimes, the pipettes are not even calibrated. Also, when you count under a microscope with the hemocytometer, it takes a lot of time to count and it's very messy. So you're not going to count a lot of cells, and that's a problem, because the calculated concentration will be based on those low numbers of counted cells. So you can imagine the variability in the results. If you count 10 cells or 1,000 cells, the precision is not the same. Another aspect is the inner operator viability. If you take people with little experience, you can see up to 100% differences between counts, which is not acceptable today for life science experiments. But even with highly experienced people, the minimum variability you can expect is 15%, which is still too much. Moreover, the dyes that you're going to use for the counts will have an impact. And when we talk about dyes in a minute, of course, cell counters can solve manual counting issues, but choosing the counter which is right for your needs requires some precautions, some steps to be sure that you select the right instrument. And Align Genetics already have 6 cell counters, and we didn't develop 6 cell counters just for the product variety. It's really because you need different technologies depending on your cells. So today we will cover some aspects, and within this webinar, I can't cover everything in details, but I will cover some aspect which seems the most important to me and make sure that you select the right cell counter. So let's start with the dyes. The most common dye and the most well-known dye is the TripandBlue. TripandBlue, as you know, it's an exclusion dye. It's a polar molecule and it doesn't enter cells if the membrane is intact. The principle with the TripandBlue is simple. You mix your cells with the same volume of TripandBlue. At the right concentration, usually which is 0.4%, the alive cells won't let the dye enter. So cells remain bright and not colored. If the membranes are damaged, it's going to stain the cell, then it's considered as dead. This is interesting because obviously it's very inexpensive, it's easy to use, you just mix the same volume of dye and cells. It's compatible with the hemocytometer or image-based cell counters, and that's perfect for easy samples like cell lines. However, with this method, this dye doesn't differentiate debris and contaminants. Debris will be dark blue, so how do you differentiate a dead cell from a debris? Other contaminants like red blood cells are dark reds. And so the only option is to count under a high magnification, meaning you're going to spend even more time counting the cells. It's not efficient and a source of errors. Anyway, therefore, TripandBlue is not accurate for complex samples. Another aspect is that this is a highly toxic compound. This is a mutagen, a teratogen, and a carcinogenic compound. So anytime you're working with TripandBlue, be careful and use gloves. If you have some TripandBlue on the surface, consider it as contaminated and decontaminate the surface. Luckily, we propose a safe alternative, which is erythrocin B. So if you're working with the TripandBlue, maybe consider switching to a safe dye. This dye is actually a food stain and it's pink colored, but it's working exactly the same way. And that is safe, and we are the only one to propose it. So TripandBlue is a common, easy to use, and inexpensive dye, but we've seen the limits and the precautions when you use it. Now, there are other dyes like the fluorescent dyes, nucleic acid dyes. The most common are DAPI, ecridine orange, and the propadiene iodide. I selected to talk about AO and PI because that's extremely relevant for cell counting. So if you look at the image on the right, this is a quite messy sample. It's a real PBMC sample from a COVID patient. If we go to count it under a microscope, we'll be in trouble because none of us would exactly count the same number of the cells. If we stain it with the AO, it's a cell permeable dye, so it means that it's not toxic for the cell. It will enter the cells and it will interact with the DNA and the RNA. Then the PI. This is a toxic compound and it doesn't penetrate the cells if the membrane is intact. Once the two dyes are in the same cell, AO will be quenched by the PI, which will turn off the signals of the AO. We have ready-to-use mix of AO-PI, so you only have to mix your cells with the AO-PI solution. But let's have a look at these two cells. Let's go step back. And so if I go back to the first image, you see that the first cell on the top, you would probably count it as a live cell, or at least a cell of an interest. This one at the bottom, you may not be sure whether this is a cell or not. Look what happens when you look at the penetration of the AO. You see the bottom cell here actually has a nucleic acid when this one doesn't have it. And PI doesn't enter the cell, enter any of those cells. It means using this kind of dyes removes the subjectivity of counting such complex samples. So in terms of advantages, AO and PI are specific to nucleated cells to nuclei. It doesn't count debris nor erythrocytes. It is compatible with all kinds of cells except bacteria. Because this is a specific situation for bacteria, but here you can count cell lines, but also more complex samples. However, you need to have the correct technology. The correct FL counter to use it. And it's not relevant if you're working with cells coming from chicken or fish, because for those species, red blood cells contain nucleic acid. So if you work on chicken or fish, there is no obvious solution at the moment. When it comes to yeast, we face another challenge. AO can be used in combination with the PI, but it's not always super efficient because of the size of the cells and the limited emission of fluorescence. So one alternative is the combination of the FDA, fluorescent, diacetate, and the PI. PI works the same way than for mammalian cells. FDA, it's a probe which can enter by diffusion inside the yeast, and the alive yeast will use their esterases to cut the FDA and to release the fluorescent. So it's an indirect method which can be used efficiently count yeast in combination with the PI. We've seen that there are different validated dyes. I didn't cover all the dyes available in the market because that would take too long, but those are the main and the most relevant dyes. Now, which technologies are able to detect those dyes and efficiently count your cells? You have the hemocytometer and a microscope. We discussed it, but it's non-efficient method. We have the cytometry. You have the possibility to use the cytometers. You're going to get much more information than only the counts. But this is highly expensive, time-consuming, and maybe you don't have access to a cytometer in your lab. Third method is the Coulter method. This is interesting method for particle counting, but far less for cell counting. This method is based on impedance measurement. It measures the changes in electrical conductance as the particles pass through a small orifice. When it goes to cell counting, a major pitfall of the technology is the price. You can go up to $4 per count, which is far too expensive. There is also lack of control. You don't see your images and it's not efficient when it comes to samples contaminated with debris or contaminants. In the market, the image-based counters are the most common and affordable solution to count cells with those viability dyes. If we say image-based counters, it's because it takes images. It takes a picture or multiple pictures. So the first technology for image-based cell counting will be the most simple ones, the most direct one. It's based on Brightfield. It takes one picture and this is really fast. It takes about few seconds. Your image then is expressed in numbers, pixels in the digital imaging processing world. You can see here in the bottom left that your cell is round. The software is going to look at the roundness of the cells and the brightness, and it will compare the pixels inside your cells, which is the foreground with the background. So if you have cells above 8 micrometers, then you will have an accurate counting. But what happens if your cells are below 8 micrometers? Problem is that the instrument, the counters, will have less pixels to compare the brightness of the cells versus the background. So if your cells are small, the viability is not always accurate. The total cell counting is accurate, but not always with the viability. With the Luna 2, we've improved the algorithm. We have the option which is called the live cell sensitivity to improve that, but still it's not perfect. And the other limit with the BF cell counters is related to the dyes you're going to use. Remember with the TripandBlue, the debris in the red blood cells are going to be counted as dead cells. So that's the limit. But if you're working with cell line or clean samples, or if your cells are about 8 micrometers, it's perfect. You're going to have ideal counting with the Brightfield image-based cell counters like the Luna 2. So here is the summary of the limits I discussed before. So if you're below 8 microns, you have an inaccurate viability, and the solution is to use a Dior fluorescence cell counters. So what are Dior fluorescence cell counters? Those cell counters included 2 technologies. You still have the Brightfield as with the previous instrument like the Luna 2, plus 2 channels of the fluorescence. So you have 3 sources of light, and here you have 2 examples of dual fluorescence instrument that we provide, Luna FL and the Luna FX7. For fluorescence cell counting, you're going to use the fluorescent dyes like the AO and the PI, which are going to link to nucleic acid. And as you can see here, it's going to count the expected cells. The red blood cells and even the platelets in the background are not going to be counted. Therefore, the cell size is not a limit anymore. It's still a fast method because it only takes 3 images in less than 30 seconds. You have your results and perfect results. And it also works with, let's say, more original samples. So if you're So I'm sorry. So if you're taking the example of chloroplasts, which are big cells, the question of the cell is not an issue. But because of chlorophyll is going to be located at the edge of the cells, it makes the count in Brightfield almost impossible, but perfect in fluorescence. So for some samples, fluorescence can solve the issue even if the size is not the problem. When it comes to bacteria, we face even more challenges. The genome size and the cell size are around or below 1 micrometer. The shapes can be like these, but it can also look like a star or even a rectangle. But you also have the challenge of the wall. Gram-positive bacteria have a thick peptidoglycan wall, and it's a problem for the dye penetration. Another challenge are related to the fact that bacteria are constantly moving. So there is a Brownian motion inside the counting slides, and they don't settle down on the same focal plane as the mammalian cells. Moreover, bacteria can be highly clustered. So you can imagine that the technology to count those bacteria is not only related to the instrument, but it also requires a bundle solution or an advanced solution. For that, we have the Quantum, a dedicated cell counter for bacteria. We optimized the loading buffer and created a specific centrifuge, an optimized dye, the instrument, cell counter itself, as well as the advanced algorithm to count the bacteria. So bacteria are very specific cells which require extra precautions. So we've seen that the combination of specific dyes and image-based technology is efficient for cell counting. The next question is it enough? Is it sufficient to ensure that your counts are reliable? The answer is no. You need to consider extra parameters. And here I selected the specific parameters that I consider as critical when choosing the cell counter. And the first point we're going to go through is the couple of definitions, accuracy and precision. Accuracy is the proximity of results to the true value. You have a target and you want to be close to that target. The precision, which is also called reproducibility, the precision is of the degree to which replicates show the same results. It's the CV of an instrument. Okay, so here this is accurate and precise. When it's accurate but not precise, you have a high CV, and this is how the result may look like. You can be precise but not accurate, which is more rare, but it also can happen. Unfortunately, what happens most is that it's not accurate and it's not precise. So what you want is accurate and precise technology, and for Logos Biosystems, it's been one of the key parameters which have been considered when developing the instrument. All our instruments have been built to be accurate and precise within the linear range of the cell counters. Here you have the Luna 2. You see that the linearity is perfect. So for cell counters, you always have a linear range. Here this is between 50,000 cells per milliliters and 10 million per milliliters, which is a nice range and perfectly compatible with your experiments. Theoretically, we could go below, but we decided to have a realistic lower limit to 50,000 cells. It means 5 to 6 cells counted. Some companies say that they can go below that, but when you count only one cell and claim that this is the lower limit, it's not statistically relevant. We go even further with the latest instrument we developed. We can go down to a CV of 1%. This is unbeatable and no other affordable solution is in the market. Not other cells counter will be this accurate and precise instrument, and whatever the consumables we're using, I'm going to show you that later, but look, we're always far below 5% of CV, and when the average of instruments are, well, for good devices, it's about 10%. For most of the instrument, Logos's products are far below 10%, but reaching 1% is a big step. So remember, accuracy and precision is critical when you select the cell counter. Another aspect is the field of view. Basically, the size of the image used to do the counting. The bigger the image will be, the more cells you will count. The more precise and statistically relevant you will be. So this is important when you make sure when you select a solution that you reach a minimum of 5 square millimeters, which represents about 0.5 microliters. And below that, there are plenty of products in the market which are minimum 30% below that. And if you consider buying such instrument, it would be better to consider keeping hemocytometers. Another question is, should I go with autofocus or not? Autofocus is important when you do routine counts. When you do lots of counts every day or every 2 days, or when you are in a production environment, because you don't want to spend time making the focus on the instrument. For R&D, it's totally up to you. It's less critical because maybe you want to look at different aspects of yourself. We propose both. For example, for the Luna 2, which is a Brightfield cell counter, it has innovative technology for autofocus, which is the liquid lens. And liquid lens has no mechanical parts in it. The principle is simple. You have 2 liquids, water and oil, which can't be mixed, and they are separated by small membrane. And when you apply the light or some current, it's going to move and adjust the focus. This liquid lens is not a mechanical part, so the instrument makes no noise in the lab. This is an innovative and patent solution you will find in this inexpensive instrument. For dual fluorescence, we have the Luna FL with the manual focus and Luna FX7 with the advanced motorized autofocus. So you have the choice. Declustering is another aspect that you need to take into consideration. Here I took the example of the first and the last instrument we put into the market. You see that straight from the first instrument, the Luna, which was our first instrument, which is currently discontinued, we have outstanding declustering algorithm. So you can see since the beginning, it was really important for Align Genetics to provide you with the best algorithm to detect the clusters. And for the FX7, the most recent instrument goes even further because most of the time, even if you're about to decluster live cells, it's highly complicated to decluster dead cells. And this instrument is capable of declustering dead cells as well. Of course, the software is important as well. The software has to be easy to use. It means that if you have to spend 1 hour or 2 hours reading the manual before using it, it's going to be complicated, and it's going to be complicated to get the best out of the instrument. The instrument has to be intuitive. But you also need advanced functions like the live cell sensitivity when you have small cells in the Brightfield. Extra options like GFP transfection, it's provided with the Luna FL. Or maybe you've measured replicates. Maybe you're interested in having the data combined directly, and that's available as well. We also have the export solution, which with our cell counters, you can export everything. You can export the images or raw images, the analyzed images, the PDF reports, which contains all the data you need, including the histograms. You can see the repartition of your cells by size. as well as the cluster map. Cluster map will tell you how your cells behave if they are alone or in clusters, and of course, consider free updates and virus-free environments. Another feature which was demanded by a number of our customers is the throughput. Most of the counters count 1 samples at a time, which is the case for most of our cell counters. But now we propose new slides which are compatible with the Luna FX7, which allow you to go to 8 samples at a time. But you can see for 8 channel slides here, we still count a reasonable volume per chamber. Remember, field of view important to have relevant results. So even with the throughput channel slides, we can bring out the relevant results as well. Last but not least, quality control. For all labs, whatever you're doing, even if you're in academics, check your instruments regularly, either with standard beats or validation slides. And if you're interested, of course, we also provide the IQOQ. If you produce cells or if you're working in GMP environments, you will be interested by the compliance, the 21 CFR Part 11 compliance. The FX7 has this option to make the counters compliant. That said, here is a question which is important, the question of pricing. You could imagine that the price depends on the onboard technology, and I strongly invite you to ask quotation to Apical Scientific. But let me answer part of this question. You see, we have a range of instruments. We have 6 cell counters. The prices will roughly go from $3,000 to $30,000. But you can see here, even with the Luna 2, which has quite advanced technology on board with liquid lens, is one of the cheapest we have. So for any quotation or pricing inquiries, please consult to our distributor in Malaysia, which is Apical Scientific. Another key aspect you need to consider is the running cost. The running cost is how much it will cost you every time you're going to click on count. So depending on the consumable you're going to use as an average, with those is going to be around 50 or 60 cents per count, which is already quite reasonable in terms of cost. But if you go with high throughput channel slides, you can decrease by half this cost. And even better, you can even pay up to 0 per count because we also have the reusable slides as well. Don't forget the quality of service and support. Ask for a demonstration, test with your own cells. We have highly educated distributors eager to support you in every single country. Ask for installation or training, even online training, because these days with the coronavirus, it can be a bit complicated to do it on site, but we can provide online training and also after sales. We've also seen many companies that say they can't repair the instrument after 1 week of warranty expiration. We don't. We repair when necessary. If your LCD screen is broken after 5 years, we can repair it at a reasonable cost. But the last word is always the customer's voice and the customer's feedback, which describes the better perception of our products. And I have to say that we're very proud these days because we received a rare label of quality for the Luna's cell counters. On Select Science, I'm not sure if you're familiar with the website, but the customers can leave the review on this website for our products. And on this website, we have reached the highest seal of quality. You can take your time to read what other users said about our cell counters. This is based on ease of use, after-sales service, and value for the money. To finalize the presentation, here is the summary slide and the last tips. If you work with cell lines, consider Luna 2. It's fast, efficient, and affordable. And if you're more oriented to R&D, consider the Luna FL and the Luna FX7. It's perfect instrument for R&D and for complex samples like PBMC. If you work with specific cells like bacteria or yeast, consider the Luna 2YF for yeast and the Quantum for bacteria. That's the end of the presentation. Thank you everyone for attending this webinar. I hope it was interesting and relevant to you.